Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this particular lecture, I will be going over digestion and absorption of dietary lipids. Now let's get into the details. Dietary lipids consist of 90% triacylglycerol. You know, triacylglycerol is a glycerol attached with 3 fatty acids of varying length. And then we'll have cholesterol ester, phospholipid, and some of the unesterified fatty acids. All these things will be there in the dietary lipids, and also along with the dietary lipids, there will be a fat-soluble vitamins, vitamin A, D, E, K. So, the digestion of dietary lipids and absorption of the simplified digested products of dietary lipids along with the vitamin A, D, E, K goes on together. Now the site of digestion of dietary lipids it begins in the stomach and it is continued to a small intestine where most of the dietary lipids are digested and the simple lipids are absorbed. So in that sense, digestion of dietary lipids begins in the stomach and that is catalyzed by two enzymes which are there in the stomach called lingual lipase and gastric lipase enzymes. So out of lingual and gastric lipase, these two enzymes, lingual lipase is the enzyme that is synthesized and secreted by sublingual glands which are there beneath the tongue, and especially the von Ebner cells in the sublingual glands are the ones that will synthesize lingual lipase and be secreted. Gastric lipase is synthesized and secreted by chief cells, and that secretion of gastric lipase, it will be stimulated by a hormone gastrin. Now both lingual lipases and gastric lipases, they are acid stable enzymes and their optimum pH for the activity is 2.5 to 6, and that is provided in the stomach, especially 2.5 to 4 pH. So that's why digestion of lipids begins in the stomach. Although we have lingual lipase secreted and synthesized in the mouth, but lingual lipase generally do not act on dietary lipids in the mouth because it needs acidic pH to be active, and that is there in the stomach. That's why both lingual and gastric lipase, since they are acid stable enzymes, so they will be active in the stomach. That's why digestion of lipids, it begins in the stomach. Now what is the target of these two enzymes? Both gastric and lingual lipases, they will act on short and medium chain fatty acids that are there in the triacylglycerol. So any triacylglycerol which contains short and medium chain fatty acids, those are the substrates for lingual and gastric lipase. In that sense, you need to know what is the carbon length in a fatty acid to call it as a short chain, and what is the carbon length in a fatty acid to call it as a medium chain. If a fatty acid has got less than 6 carbons, it is a SCFA, short chain fatty acid, and the medium chain fatty acid has got a carbon between 6 to 12 carbons. So any triacylglycerol that is attached with short chain fatty acid and medium chain fatty acid, they are the substrates for lingual and gastric lipase. For that matter, remember, any enzyme that ends as lipase, the substrate for lipase is triacylglycerol. Remember that. Any enzyme that you come across which ends as lipase, it is the substrate for that lipase is triacylglycerol. Anyway, now other than this lingual and gastric lipases, there is a mammary lipase which is synthesized and secreted by lactating mammary glands. And the mammary lipase is not inactivated by acid present in the stomach, and it will be acting on long chain fatty acids which are present in a triacylglycerol that is synthesized and secreted by lactating mother. Now, these are the three lipases that we have seen now so far: gastric lipase, lingual lipase. These are acid stable enzymes. Lingual lipase is coming from the lingual glands beneath the tongue in the mouth. Gastric lipase is coming from chief cells of the stomach stimulated by gastrin, and mammary lipase is coming from lactating mammary glands. Now also know that mammary lipase, it is specific for long chain fatty acids, and you should know what is the carbon length in a long chain fatty acid. Long chain fatty acid has carbons more than 12 to less than 22. That means from 13 to 21 carbons, that is belong to long chain fatty acid. Lingual and gastric lipases, they play an important role in lipid digestion in the infants and young children, and also those with pancreatic insufficiency. It's because when milk is the energy for infants and even in young children, lingual and gastric lipases are the ones that can be useful in the digestion of short and medium chain fatty acid containing triacylglycerol present in the milk and digested in the stomach. And also when a patient is having a pancreatic insufficiency, example cystic fibrosis, where a pancreatic secretions are not coming into the duodenum for digestion and absorption process of dietary lipids, proteins, carbohydrate. So these patients, they won't be able to digest lipids, especially the triacylglycerol containing long chain fatty acid. In that time, lingual and gastric lipases, they become important to digest a dietary lipid, especially that contains short and medium chain fatty acid containing triacylglycerol. Once a partially digested dietary lipid gets into a duodenum, when the acidic content gets into the duodenum, so in response to the acidic content that gets into the duodenum, there will be release of two hormones from the duodenal glands, and those two hormones are cholecystokinin and the other hormone is secretin. Both cholecystokinin and secretin, so what they do is, cholecystokinin, it brings pancreatic enzymes, whereas secretin will bring bicarbonate. So the pancreatic enzymes, they will participate in digestion of dietary lipids, proteins and carbohydrate, whereas bicarbonate is going to neutralize the acidic content that got into the small intestine from the stomach. So now we are looking at digestion and absorption of dietary lipids in this chapter. So we will restrict ourselves with understanding how dietary lipids are digested and absorbed, rather than going into or deviating into how dietary carbohydrates and proteins are also absorbed. We are going to learn them separately. Now, the lipids are there in the small intestine coming from the stomach, partially digested by the action of lingual and gastric lipase or mammary lipase. Now, lipids are hydrophobic, insoluble in water, so all they will coalesce and make a lipid mass. So now, in order to digest all this lipid which is hydrophobic, so what happens is, there will be breakdown of this large lipid mass by the intestinal contraction into small lipid droplets, while these lipids are coated with bile acids and bile salts, because bile acids and bile salts, these are amphipathic molecules and they act as a detergents, and thereby they help in breaking of large lipid molecule into simple lipid droplets and keeps their association with that small little droplets. Alright, so what type of bile acids and bile salts we have? So one is a glycocholic acid or taurocholic acid, other type of bile acid is 𝐒𝐵𝐾 acid, 𝗠𝐴𝐶. These bile acids, they have got low pKa. That means they will undergo deprotonation in a pH of intestine which is around 6 to 6.5. That means the deprotonated form of these bile acids, they will retain that amphipathic character and they will go and bind with the lipids on their hydrophobic side, and their hydrophilic side will be towards the water environment in the intestine. And when this coating of lipid globule with the bile acids and bile salts occurs, and when the peristaltic action breaks lipid globules into small lipid droplets, which we refer them as micelles. So collectively, all these small little lipid droplets, they increase the surface area of the lipid. So increased surface area will allow rapid lipid digestion by digestive enzymes coming from the pancreas. So that is what is shown here in the figure. So as you can see, lipid, which are a big molecule here, hydrophobic, it will undergo breakdown process into simple lipid droplets, and these simple lipid droplets are coated by bile acids and bile salts. So as I was saying, this bile acid, it is an amphipathic molecule, so it has a polar head and it has a non-polar tail. And the non-polar tail, it is going to interact with the lipid side in the interior, and the polar head is outside where water molecule is there in the lumen of the intestine. Okay, like this, the bile acids having amphipathic nature, so they will keep these lipid droplets kind of soluble in the intestine, and also breaking a big lipid molecule into small lipid droplets, they will increase the overall surface area collectively and help lipid digestive enzymes coming from the pancreas to attack all these molecules simultaneously and increases lipid digestion process in a short time. Okay, and that is what is the purpose of having bile acids and normal intestinal contractions to break the large lipid molecule into simple lipid droplets, which we refer them as micelles. Okay, this entire process we call it as emulsification. So the emulsification is coating of lipids by bile acids and bile salts and continued peristaltic action, where a micelle formation is going on, and these micelles containing lipids, or they retain their association with the lipids with the bile acids and bile salts, where pancreatic enzymes will go and work and digest lipids present in them. Okay, and so this is where the bile acids and bile salts role help. So basically they assist or they enhance the lipid digestion process in the intestine. Now, what are the types of pancreatic enzymes that we have? So the pancreatic enzymes are released by cholecystokinin hormone, and these pancreatic enzymes, they come from the pancreas and transported all the way to the second part of duodenum by a common pancreatic duct through ampulla of Vater. Now the secretin brings bicarbonate to the intestine, neutralize the pH there. Now the pancreatic digestive enzymes that come from pancreas related to lipid digestion are pancreatic lipase. Pancreatic digestive enzymes that come from pancreas for lipid digestion process are pancreatic lipase, pancreatic procolipase, pancreatic cholesterol esterase, pancreatic phospholipase A2. Now procolipase has to be converted into colipase. So procolipase is an inactive form of an enzyme. So that particular activation process, it will be done by enzyme trypsin. And note that this trypsin as such, it will come from pancreas in its inactive form, zymogen form, as trypsinogen, and the first trypsinogen has to be converted into trypsin, and that is done by an enzyme called enterokinase or enteropeptidase, which is attached to the brush border epithelium of our intestine towards the luminal side. Once trypsinogen is converted to trypsin, and that trypsin is going to activate most of the zymogen enzymes coming out of the pancreas, and one of the enzyme which is in the zymogen form participating in the dietary lipids digestion is a pancreatic procolipase, which is activated into colipase, and this colipase, it works along with the pancreatic lipase to digest triacylglycerols into 2 free fatty acids and 1 molecule of 2 monoacylglycerol. Now, the pancreatic lipase do not have any specificity related to the number of carbons that are attached to a triacylglycerol. Only specificity for pancreatic lipase is it can only work on first and the third carbon attached fatty acid in a glycerol molecule, but not to the second carbon that is attached to a glycerol molecule. So, the second carbon fatty acid is not released by the pancreatic lipase. So, that is why we get 2 free fatty acids and 1 molecule of 2 monoacylglycerol upon the action of pancreatic lipase along with the colipase. Then why do we need colipase actually? The colipase it helps in the activity of pancreatic lipase because bile acids and bile salts have an inhibitory effect on pancreatic lipase, and having colipase together, that inhibitory effect is relieved. That's why pancreatic lipase needs pancreatic colipase. Now, there is a drug called orlistat. Orlistat inhibits gastric lipase and pancreatic lipase, thereby it decreases digestion of dietary triacylglycerol, and thereby triacylglycerols are complex molecules. They are not absorbed by the intestinal cells. It is simply going out in the feces, and thereby decreases triacylglycerol levels in the blood. That's what is the mechanism of orlistat, which is used as one of the anti-obesity drug. Now, you can see the action of pancreatic lipase in this particular diagram. So, 3 fatty acids attached to a triacylglycerol. The third carbon fatty acid is released as free fatty acid, now making a diacylglycerol, and diacylglycerol releasing 1 more fatty acid at the third carbon, becoming 2 monoacylglycerol. And this 2 monoacylglycerol and 2 fatty acids, so basically the outcome here after the action of pancreatic lipase along with colipase, so we get 2 free fatty acids and 1 molecule of 2 monoacylglycerol. 2 free fatty acid and 1 molecule of 2 monoacylglycerol. Now, these 2 monoacylglycerol and 2 free fatty acids, they retain their association with the bile acids and bile salts. Okay. So, we are going to see what exactly the molecule called later. Now, sometimes what will happen, this 2 monoacylglycerol can be isomerized by 2 monoacylglycerol isomerase enzyme into 1 monoacylglycerol. And now this 1 monoacylglycerol can be acted on by pancreatic lipase, because pancreatic lipase can break fatty acid attached to the first carbon. So, we are going to get a free fatty acid plus a glycerol molecule. That's how triacylglycerol can be completely degraded into 3 free fatty acid and a glycerol molecule. That's one fate of it. The second fate can be 2 free fatty acids and 1 monoacylglycerol molecule. 2 monoacylglycerol molecule. Now, coming to other pancreatic digestive enzymes, that is pancreatic cholesterol esterase enzyme, which is going to hydrolyze a cholesterol ester and release free fatty acid and a cholesterol molecule, and bile salts enhance this reaction. You can see that here in the figure. Cholesterol ester is converted into cholesterol plus a fatty acid is released, done by pancreatic cholesterol esterase. Similarly, a phospholipid. It is acted by pancreatic phospholipase A2 enzyme, releasing a lysophospholipid and a free fatty acid. So like this, dietary lipids are digested into free fatty acids, free cholesterol and lysophospholipid. And all these molecules, let me write it down. Free fatty acids, cholesterol, lysophospholipid and vitamin A, D, E, Ks. All of them, they retain their association with bile acids and bile salts. So bile acids and bile salts are kind of coating them, and these fatty acids are inside because of their hydrophobicity and all that. So they retain their association with their bile acids and bile salts, and these kind of molecules, we call them as mixed micelles. So basically, micelles are converted into mixed micelles in the intestine. So what are micelles? Micelles, as we have seen earlier, in the initial stages of lipid digestion and absorption, bile acids and bile salts are coating a dietary lipids which contains diacylglycerols, phospholipid, cholesterol, ester. These are all undigested lipid molecules, very complex. So bile acids and bile salt coating, and then they break into small lipid droplets and they retain their association with the bile acids and bile salts. All that undigested lipid containing molecules, we call them as micelles, and the molecules that contains digested lipids which retain their association with bile acids and bile salts, they are called as mixed micelles. And mixed micelles usually contains long chain fatty acids. Why they don't contain short and medium chain fatty acid? I will come to that point. They contain long chain fatty acids, 2-monoacyl glycerol sometimes, lysophospholipid, cholesterol, along with that vitamin A, D, Ks. Now coming to the short and medium chain fatty acids. Short and medium chain fatty acids are not being part of the mixed micelle because short and medium chain fatty acids being water soluble, they are absorbed directly into the portal blood. Only the long chain fatty acids and other molecules, they are part of the mixed micelle, and now they are ready for absorption. Now let's get into the absorption of digested lipids from the enterocytes. The bile acids which are coating mixed micelles with their hydrophilic layer outside. So these hydrophilic part of the bile acids, they are going to interact with the water molecule present over the enterocyte surface and remove that water molecule, remove the influence of water molecule, or remove that water layer by the bile acids and allow the free diffusion of these digested lipids inside the enterocytes. Now let's look at absorption of lipids by the enterocytes. So as you can see here in this particular figure, that the brush border epithelium of enterocyte, it is coated by the water layer because it is there in the lumen of the intestine. There is a lot of water there. So the water coating will be present in the outer surface. Now the mixed micelles which contain digested lipids like free fatty acids, then lysophospholipid, cholesterol, long chain fatty acids, all of them need to undergo free diffusion through the brush border epithelial membrane. So there is a water layer which is coating this brush border epithelium, as I am highlighting with the green color, and that water layer need to be removed first, and that will be done by the hydrophilic head of bile acids that are coating the lipids. So as you know, Bile acid has got a polar head and non-polar tail. So the polar head is going to interact with water and it removes that water layer, thereby allow the free diffusion of most of the dietary lipids. And that's how the dietary lipids undergoes free diffusion. So that means free fatty acids, 2-monoacylglycerol, lysophospholipids, vitamin A, D, Ks, all of them, they undergo a simple diffusion inside the enterocyte. Whereas cholesterol is a poorly absorbed molecule and that needs a facilitated transport mechanism, and that is done by a transporter called NPC1L1. And what is NPC1L1? NPC1L1 is Niemann-Pick C1 and like 1. That is the name of the transporter. And this NPC1L1, it is inhibited by a drug called ezetimibe. So ezetimibe is an inhibitor of NPC1L1 and it is used in the treatment of hypercholesterolemia. Okay, so like this, lipids are digested and they are absorbed. Most of them are absorbed by simple diffusion and cholesterol is absorbed by NPC1L1 transporter. Now what about bile acids and bile salts? Bile acids and bile salts, after they assist in the absorption of dietary lipids, so they will move on to the terminal part of ileum, and where in the terminal part of ileum, so they are reabsorbed into the portal system from where they are carried to the liver. So more than 95% of the bile acids and bile salts are reabsorbed. Only less than 5% of the bile acids and bile salts are lost out of our body in the feces. Okay, now once lipids are absorbed into the enterocytes, so they are directed towards the smooth endoplasmic reticulum. So now in the smooth endoplasmic reticulum, these long chain fatty acids are activated into fatty acyl-CoA by fatty acyl-CoA synthetase enzymes, and activated fatty acyl-CoAs, they are used for esterification process. What kind of esterification going on here? So the 2-monoacylglycerol that is coming from triacylglycerol digestion will be added with 2 free fatty acids as it is shown to become a triacylglycerol. Similarly, cholesterol is added with a fatty acid to make a cholesterol ester, and phospholipid, especially coming in as lysophospholipid, are esterified to become a phospholipid. All these reformed lipids into their complex form, back into their complex form, like triacylglycerol, cholesterol ester, phospholipid, they all need to be transported out of the enterocyte into the circulation. Now my question is, why there is a reformation of triacylglycerol, cholesterol ester and phospholipid when in the intestinal lumen they were broken down into simpler form? The reason for that breaking or digesting the triacylglycerol, cholesterol ester, phospholipid into their simpler form is to allow the absorption process to go on, because in the complex form they are not absorbed. So they need to be simplified. Once they are absorbed, so they will have to be brought back into their complex structure, the original structure. And now all these complex molecules which are made from the reesterification process along with vitamin A, D, Ks, they all are present in the smooth ER and they will be transported to a rough endoplasmic reticulum where a protein called ApoB48 is present. And ApoB48, it is going to receive all these lipids, and with the help of ApoB48 there is a formation of a complex molecule with all the triacylglycerols and cholesterol ester hydrophobic molecules inside and phospholipids and free cholesterol outside, with the apolipoprotein B48 and that entire structure we refer it as chylomicron, and the chylomicron is a big molecule, so it needs bigger fenestrations to enter into the circulation. That's why it preferred to enter into lymphatics, lacteals, and from there it will find its way into systemic circulation, and that's what happens to dietary lipids that are absorbed. Now this figure here, it is giving you overview of all the information that I have explained so far, starting from the digestion of dietary lipids in the lumen of the intestine, then the absorption, and then the transport process. So just to quickly revise that, let me just highlight some of the important points. So in the intestinal lumen we have triacylglycerol, cholesterol ester, phospholipid, vitamin A, D, Ks, all of them are coming in. So here in this figure it's showing only triacylglycerol, but our food, if it contains other like phospholipids, cholesterol ester and all that. So the pancreatic lipase along with the co-lipase digest triacylglycerol into 2-monoacylglycerol and 2-monoacylglycerol further, and 2-monoacylglycerol and 2 free fatty acids released. 2-monoacylglycerol further isomerized into 1-monoacylglycerol, and that is further digested by pancreatic lipase into free fatty acid and glycerol, and glycerol just directly goes into the portal circulation after absorption. Short and medium chain fatty acids also go into portal circulation after absorption through enterocytes, whereas other free fatty acids containing long chain fatty acids and then cholesterol, cholesterol ester, phospholipid, all of them, they need to be re-esterified in the smooth endoplasmic reticulum. Once they are re-esterified, they will be loaded on to ApoB48 along with vitamin A, D case, and that makes chylomicron, and chylomicron is secreted into lymphatics, and that's how lipids are carried to the systemic circulation from the intestine. So that is what I have written here in the figure. Chylomicrons are secreted by the enterocytes and transported in the lymphatics and then find their way into the bloodstream. In the bloodstream, tags in the chylomicrons are degraded and utilized by the peripheral tissues in chylomicron metabolism. So the secretion of lipids from the enterocytes shown here in the form of chylomicrons, where smooth ER, where the lipids are present. In the rough ER we have apolipoprotein B48. So once the things are loaded, like the chylomicron which is formed there, it will be diverted into Golgi complex, and the Golgi complex makes a secretory vesicles, and chylomicron is exocytosed into lymphatics rather than portal capillaries. The reason for that is lymphatics fenestrations are much bigger than the portal capillaries. Now coming to the regulation of lipid digestion process. So let's use this figure here to understand the regulation process. So we have two hormones, cholecystokinin and secretin. So the cholecystokinin, it is going to bring pancreatic enzymes, especially the exocrine pancreas, releasing pancreatic digestive enzymes. Along with that, cholecystokinin, what it does, it is going to inhibit gastric motility, thereby gastric contraction is not going on. So thereby it allows a sufficient time for the small intestine to digest the lipid that has already reached small intestine coming from the stomach. Now what secretin does? Secretin, it is going to bring bicarbonate and neutralize the acidity or acidic content coming from the stomach. So like this, both cholecystokinin and secretin, they help in digestion of dietary lipids. So with a recap that I have written here, in response to acidic content, cells of duodenum releases cholecystokinin and secretin. Cholecystokinin causes contraction of the gallbladder and brings bile acids and bile salts for digestion of dietary lipids. Cholecystokinin releases pancreatic enzymes, and also cholecystokinin, it decreases gastric motility, whereas secretin brings bicarbonate from the pancreas. So all these processes will help to digest dietary lipids in the small intestine. Now coming to lipid malabsorption disorders. A condition called steatorrhea, in fact it is a sign called steatorrhea. It simply indicates fat in the stools is much more than 6 grams per day. It is an excess presence of fat in the feces. That is what is called as steatorrhea. So during steatorrhea, stools will have oily appearance, difficult to flush, and also foul smelling. So what are the causes for steatorrhea? Any interference with the digestion and absorption of dietary lipids will lead to steatorrhea. That can be lack of bile acids, because bile acids are needed for emulsification process. So lack of bile acids can be intrahepatic or extrahepatic bile duct obstruction, and the example for extrahepatic bile duct obstruction is a stone in the common bile duct or cancer in the head of the pancreas. Intrahepatic bile duct obstruction can be primary sclerosing cholangitis or disorders which are affecting the liver. Exocrine pancreatic insufficiency, where pancreatic juice is not coming into the duodenum for digestion, like example cystic fibrosis condition or pancreatic duct obstruction itself, or any diseases affecting the small intestine can also lead to lipid malabsorption. Can be celiac disease, tropical sprue, bacterial overgrowth. All of them can lead to lipid malabsorption. So in the lipid malabsorption, where the necessary things are not reaching the intestine, necessary things like bile acids, pancreatic juice or bicarbonate, or the mucosal damage, defective cells in the small intestine, all of them will lead to dietary lipids not being absorbed properly and allow that lipid to get out of our intestine in the feces, causing steatorrhea. Now in steatorrhea, there will be deficiency of fat soluble vitamins, vitamin A, D, K, and essential fatty acids. So this can be seen in conditions like obstructive jaundice or cystic fibrosis or intestinal malabsorption of lipids because of damage to the mucosal membrane or inflammation of the intestinal mucosal membrane. So note that. Generally in lipid malabsorption, we don't find a triacylglycerol deficiency, phospholipid deficiency, cholesterol deficiency, because these are the molecules that can be synthesized in our cells. They are not absolutely essential in the diet. Whereas fat soluble vitamins other than vitamin D and vitamin K, like especially vitamin E and A, so they are absolutely essential because we cannot synthesize them. Essential fatty acids like linoleic acid and linolenic acid, they are absolutely necessary for us and need to be taken in the diet, and in lipid malabsorption they can be deficient. One of the artificial fat substitute that we use in order to decrease dietary lipids in people with overweight, obesity, who want to avoid fatty food but still have the taste of the fat, in that condition we use a molecule called olestra. Olestra is an artificial fat substitute. It consists of sucrose molecule to which fatty acids are esterified to hydroxyl group, and the fatty acid attached to sucrose is resistant to hydrolysis by pancreatic lipase enzyme. So in the mouth, sucrose portion of the molecule imparts a sweet taste. Olestra causes deficiency of essential fatty acids and fat soluble vitamins because it contributes to lipid malabsorption. So olestra are supplemented with fat soluble vitamins and essential fatty acid. So here is the structure of olestra, where sucrose is attached with the fatty acid. So this is all about digestion of dietary lipids and absorption of simple digested lipid molecules. I hope this video has helped you in understanding lipid digestion and absorption process. Thank you.